little bit of time to go through some of the material from Chapter 9. And this first little section here about how microbes grow, I just want to cover a little bit about binary fission. So binary fission is the way that bacteria replicate. And, you know, when we were looking, I guess it was lab 3, when we were looking at some of our bacterial shapes, we could see how some, you know, were still connected together. They hadn't, you know, fully separated. And that is because the binary fission had not completely happened yet. So basically what happens, you can see here in this diagram, first thing that happens is the DNA is going to replicate. The cell is going to make more ribosomes, more cytoplasmic filaments, more components of the cell wall, and so the cell is going to get bigger. And then you get, you know, as the cell is elongating, as it's getting bigger, you get this septum that starts to form, kind of this little furrow that forms on either side. And eventually what's going to happen is the DNA is going to separate, and then the cell is going to separate into 2 new cells. Now, these are clones of one another, meaning they're the exact same cell. They have the exact same genetic material. And so what's going to happen then is these cells then are going to be able to divide. And the dividing time depends upon the species. So how long this process takes is going to be dependent upon the different species of bacteria. Okay. And that is called the generation time, or the doubling time. So you see here, you know, they give us the example, E. coli can double in as little as 20 minutes under its optimal growth conditions. So what that means is that 1 can become 2 in 20 minutes, and then 2 becomes 4, and then 4 to 8, and 8 to 16, and so on and so on and so on. That's why we call that the doubling time. It's the time it takes for the population to double. Most pathogens are going to grow pretty rapidly. You know, that's why we, you know, put our things in the incubator for 48 hours and we come back and we have a lot of growth that we can observe, because the doubling time is fairly short, somewhere between, you know, 20, 30, 40 minutes. Some, like you see here, Mycobacterium tuberculosis, generation time of 15 to 20 hours. So it grows a lot slower. And that we do see that with some other bacteria. Now, when we look at bacterial growth curve, this is what we see. Okay, this is a logarithmic growth. And so what happens is, at first, when we put the culture, when we get ready to subculture and we introduce that culture into a new tube or into, a new growth medium, we have what's called the lag phase. And this is just the time it takes for them to get used to their new environment. Okay, so they get introduced to the media, they get put in the incubator. At first, nothing happens because they just need time to get introduced to their new environment. And then you see here we get what's called the log phase. And the reason why we call this a log phase is because it can be calculated logarithmically. Okay, so we can actually do mathematical calculations to determine what our increase is going to be. And so we just basically get this explosion of growth. Okay, then we get to what we call the stationary phase. And in the stationary phase, we get the rate of cell division and the rate of cell death are equal. So basically what's happening is you get a plateau, because the cells are still dividing, but now some of the cells that were, growing in that log phase are starting to die off. So we don't get a net increase in the number of organisms. And then eventually we get to the death phase. And now the number that are dying exceed the number that are being reproduced. And typically what happens is waste products have started to build up. You know, whatever waste products the bacteria are giving off, whether that's some type of acid or it's, you know, carbon dioxide or whatever it may be, those waste products are starting to build up. They're starting to run out of oxygen. They're starting to run out of the nutrients. You know, when we put the bacteria into the media, it has proteins and it has sugars and it has, you know, different growth requirements in there. Well, just like with any living organism, if they remain there for too long, all of their nutrients are going to be used up. Okay. So we get to this death phase. So this is the growth curve of bacteria. Lag phase, log phase, stationary phase, and then we get to that death phase. And if no new nutrients are added, then we're going to just continue in that. Okay. And so basically this graphic kind of shows you, like, if we're going to sustain the bacterial growth, then we have to be able to pull off those waste products. We have to be able to introduce more nutrients in there. Okay. Now, we don't really do an actual measurement of bacterial growth. You know, there is a way we can do cell counters. There is a way to do this. We don't do this. We don't do an actual cell count. Really, what we do is when we look at our tubes, you know, and I've mentioned this several times, that we talk about the turbidity, we talk about the cloudiness, you know, and that's a measure of how many bacteria there are. So like this here, it's an indirect method. Okay, the turbidity, how cloudy is the tube. Okay. Not going to go over biofilms. You've watched a couple of videos about that. So now we're going to get into the oxygen requirements, and this is what we're going to work on in lab next week. So we can look at the oxygen requirements for growth by using a particular type of media called thioglycolate or fluid thioglycolate, and that's what we're going to use. The thioglycolate medium basically has very little oxygen. So when we put the bacteria in, we can get this growth pattern that results based on the bacterium's need for oxygen. Okay. And we briefly talked about this a little bit when we talked about the Catalase test, but when we look at oxygen requirements for bacteria, we have basically 5 different categories, I guess you could say. So we have obligate or strict aerobes. Obligate aerobes are those that must have oxygen to survive. So we are obligate aerobes. We must have oxygen to survive. Without it, we die. Okay? And so that we have a lot of bacteria who are obligate aerobes. They must have oxygen or they'll die. Then we have those that are the complete opposite, which are the obligate anaerobes. With obligate anaerobes, they are killed by oxygen. So they do not want to be exposed to oxygen. Okay. Then we have the facultative anaerobes. Facultative anaerobes prefer oxygen, but they can grow in the presence or in the absence of oxygen. Okay. So they prefer oxygen, but they can switch over to a fermentation type metabolism or an anaerobic metabolism. So they can survive without oxygen, but it's not sustainable for a long period of time. Okay, so they prefer oxygen, but can do without it for short periods of time. Then we have the Aero Tolerant. Okay, Aero Tolerant anaerobes. And you see there, it says they're indifferent. They don't use it, but it doesn't hurt them. So they could care less. They're indifferent. They don't need it to survive, but it's not going to harm them like it does the obligate anaerobes. And then we have the micro aerophiles, and your book refers to these as the Goldilocks. You know, it's got to be just right, not too much oxygen, not too little oxygen, about 1 to 10%, which is less than our 21% in the atmosphere. So atmospheric oxygen is too much. So they need smaller amounts of oxygen. And so this is what the tubes are going to look like. So the obligate anaerobes, you see they're going to be clustered up to the top because, you know, there's going to be that gap where there's some oxygen there. And so the obligate aerobes are going to be up at the top so they can take advantage of that oxygen. The obligate anaerobes, they're going to want to be as far away from the oxygen as possible. So they're going to be down close to the bottom of the tube. Facultative anaerobes, we'll see more up at the top, but then we can kind of see them throughout. The Aero Tolerant, again, they don't care, so they can really be anywhere, okay? And then when we look at the micro Aerophiles, they're not at the top because that's too much oxygen. So they're somewhere in the top third or so, okay? So this is one of the labs that we're going to do next week, and we're going to get our results and then try to classify our bacteria based on where we find them in the media. Okay. All right. Now, we're not able to do this in our lab, but you see here this image of what we refer to as an anaerobic jar. An anaerobic jar is where we're able to grow those obligate anaerobes. We can't grow them in normal equipment because they're going to be exposed to oxygen and they'll end up they'll die. And so basically what happens is this would be vacuum sealed, essentially, to pull all of the oxygen out of the environment. Okay. And we talked about the catalase before. You know, catalase is the enzyme that helps to break down oxygen, essentially. And if an organism has catalase, then that means they can tolerate the oxygen. If they don't, then they can't. Now, just like oxygen, pH has an effect on microbial growth as well. And we're not going to do any labs that look at pH at all. But this section just kind of talks about why pH is important. So most all living organisms grow best at a neutral environment. Okay, you see there, most bacteria are what we refer to as neutrophiles. They grow in a neutral environment. So neutral is 7, so they're going to be somewhere between 6 and 8. Okay. However, when we look at some of the pathogens that affect the stomach, when we look at fungi, they like to be, you know, on the slightly acidic side. And so we still refer to them as neutrophiles, even though they're a little bit lower than 6. Our acidophiles, we use that term for those that grow, you see they're less than 5.55. So if we get to about 5.5 and below, then we call those acidophiles. They like to be in an acidic environment. And you see there, we find some at Yellowstone National Park. And most of these are archaeans. They're not in the domain Eubacteria. They have adaptations that allow them to survive in that environment. They're tolerant of the acidity. A lot of times they can pump the hydrogen ions, which is what makes the environment acidic. They can pump those out of their cells so that the acidity doesn't bother them. And then we have the alkaliphiles or the alkalinophiles, and they grow best between 8 and 10.5. And you see there's a neat picture here of Natronobacterium found in the African Rift Valley at 10.5. And so they kind of show up pink there, which is kind of kind of a neat thing. So those are the alkalinophiles. They thrive at higher pHs. So again, we're not going to work with any of those in the lab. Now, we will do a lab involving temperature. So when we look at temperature, all organisms have an optimum growth temperature. And anytime you see their optimum, that's where they're growing at their best rate. So when we talk about that logarithmic phase, we get the fastest amount of logarithmic growth at an optimum condition. So when we're talking about temperature, we have an optimum temperature. And then we have a minimum temperature and a maximum temperature. Okay. Now, the organisms that we work with for the most part are mesophiles, okay, or middle loving. They're in the moderate temperatures with room temperature to about 45 degrees. So pretty much all pathogens are mesophiles because body temperature is 37 degrees Celsius. So pretty much all pathogens in our normal microbiota, they are mesophiles. Now, we have other organisms called Psychrotrophs. Okay, now phile means love. Troph means basically eating, really. And so the Psychrotrophs basically tolerate the temperature. They don't love the temperature. Okay, so Psychrotrophs or Psychrotolerants prefer cooler environments, so about refrigeration to about room temperature. So we find these, out in the environment. This is what, can cause food spoilage, things like listeria, for example. Listeria is a psychrotroph. But then we get to a class below those to the psychrophiles. Now, psychrophiles, you see there, their optimum growth temperature is about 15 degrees Celsius. So that's even cooler than average room temperature, and they can grow at 0 degrees and below. These are found, in the Arctic and the Antarctic. They're very important in decomposition processes at like the North and South poles in the tundra. Then we have thermophiles and hyperthermophiles. So thermophiles are about 50 degrees Celsius to 80 degrees Celsius. And then the hyperthermophiles from about 80 to 110 degrees Celsius. You see there's some that survive above 121 degrees Celsius. So we're talking here, well above boiling water temperature. Okay. Again, these hyperthermophiles most of the time are archaea. Okay? They don't belong in the eubacteria domain. Here's a picture of a vent at the bottom of the ocean where we would have bacteria, these hyperthermophiles that would live there. Okay. Now, when we talk about these hyperthermophiles, these acidophiles, these alkalinophiles, a lot of times you'll see them referred to as extremophiles because they live in these extreme environments where a lot of things really don't live. And so that is why we refer to them as extremophiles. Now, something else that affects growth, and again, something that we can't test in the lab, is osmotic pressure or barometric pressure. So when we refer to osmotic pressure, we're referring to the halophiles. Now, halo or halo means salt, and so the halophiles are those that love salt. And it's not so much that they love salt, it's that they love the pressure that's created by a salty environment. So to kind of give you an example of this, so when you see slugs, right? And so to get rid of slugs, you pour salt on them. And salt basically dissolves them, or it kind of looks like it dissolves them. It makes them dry out. So what happens is the salt creates an environment that draws water out. And so when you pour salt on the slug, the water from the slug cells are going to leave the slug, okay? Because it wants to get to what we refer to as an isotonic environment, okay? It's the same idea of why when you go in the hospital and they give you an IV, they give you saline. They don't give you pure water because they want to match the salt content of our cells, of our blood. So they give you what we refer to as normal saline. So halophiles live in areas where there is a high salt concentration. You see they're found in marine environments like in the sea where salt concentrations hover at 3.5%. We have those that live in the Great Salt Lake or in the Dead Sea. They require that osmotic pressure, that hyperosmotic environment in order to survive. If you were to put them in just a normal saline environment or in normal water, they're going to die. They're not going to be able to survive that. And so these halophiles can withstand environments that other bacteria can't. And so if we look at this picture here, this is from the Great Salt Lake. And you see there you get this kind of purplish color. This is due to a particular alga that lives there, and also the Halobacterium genus. And again, these are archaea. Okay, this Halobacterium, this is an archaea. And a lot of our archaea are going to be these extremophiles. Okay, they're going to live in these extreme environments. Okay. Now we have other bacteria that we refer to as halo-tolerant. Okay. They don't require that very high osmotic pressure created by the salty environments to survive, but they can survive it. And one of those we're going to work with in the lab week after next. Staphylococcus aureus is a halo-tolerant organism. Well, really, multiple staph species are. They can tolerate salty environments. Okay. Now, another group of these extremophiles are referred to as the barophiles. So baro means pressure. And so these organisms can withstand extreme pressure. So we find these essentially at the bottom of the ocean. Okay. You know, the deeper you go in the ocean, the more and more and more atmospheric pressure is being pushed down on you. And so these bacteria live basically on the ocean floor. And if we were to try to bring them up, they would not survive. They need that high barometric pressure. Okay. So we are going to use some of these in this 9.6 here, some of the different media that are used. And we're going to use McConkey, and we're going to use mannitol salt agar. We're going to use blood agar. We're going to use the thioglycolate media next week. So we get into these specialized media, and there are hundreds of them. I mean, we could spend, you know, a whole 16-week semester just looking at the different types of specialized media there are. So, you know, we don't get into all of that, but we do use a few of these so you can see these different forms of bacteria. We're going to do one on temperature. We're going to do one on oxygen. We're going to use the mannitol salt agar to look at these halo-tolerant species. We're going to look at the blood agar for those that need, they basically feed on red blood cells. And we use one that's tryptic soy agar with sheep blood in it. So we do get to use some of these other forms of media. So I hope that helps kind of go over some of the things that you've been reading and that we're going to look at in the lab over the next few weeks. If you still need to make up labs, we'll discuss that this coming week. Probably have you come in early on Tuesday, but I'll take care of all of that when I get back.